Hello everybody, this is chapter 9, muscles and muscle tissue. The purpose of this chapter is to discuss skeletal muscle. We'll start off with looking at its anatomy, and then we'll discuss the neuromuscular junction and the sliding filament theory. Towards the end of the chapter, we will have a discussion also about smooth muscle, where we'll take a look at its contractile units, and obviously when we compare it to skeletal muscle, what do they have in common and what sets them apart from each other. Before we get into more of a detailed discussion, let's do a quick comparison of our three muscle types. So in your histology chapter, you learned that there are three types of muscles. We have our skeletal, our cardiac, and our smooth muscle. Skeletal muscle is known for being a striated muscle type, and that's because from a histology point of view, if you stick it under the microscope, you're going to see perpendicular lines running through its muscle fibers. These perpendicular lines are going to be the contractile element of the muscle. Skeletal muscle is usually seen with attachment points to the bones, as well as to the skin, and we know that it's under voluntary control. The second muscle type is going to be cardiac muscle. This one derives its name from the fact that it's only located within the walls of the heart. It is also a striated muscle type, and in fact, when we look at the histology, we're going to notice that some of the striations are brighter colored than others, and that's because those are the intercalated discs, the meeting points between two cardiomyocytes, two little heart cells. Cardiac muscle is under involuntary control, meaning that you cannot control it on a conscious level. The third muscle type is going to be smooth muscle. Smooth muscle is an unstriated or a non-striated muscle because the contractile units are not perpendicular aligned. It is under involuntary control, and when we look at its location, we're going to find it in a lot of our visceral or internal organs, and we'll also find it in things like arteries and in our airways, and it's also been found in some of the muscles that have to do with our eyes. Muscle tissue is also very unique, especially when you compare it to epithelium and connective tissue, because it's going to have these four characteristics that set it apart from the rest. It will be excitable, it will have a contractility characteristic, an extensibility characteristic, as well as an elasticity component. Now, excitability relies on the fact that the muscle can go ahead and be stimulated and it will respond accordingly. So in a little bit, for instance, we're going to talk about the fact that if you take an electrical stimuli and you go ahead and you allow it to pass down an axon of a neuron, that neuron will be able to stimulate a skeletal muscle, and if the threshold is met, the skeletal muscle will go ahead and contract. That will be its response to the stimuli. The fact that it can have a contractility means that it can shorten when it's stimulated. If it's extensible, that has an extensibility, that means it can be stretched or elongated. And elasticity basically means that it can go back to its original or resting length. On the right-hand side of the PowerPoint, it highlights some of the important functions that we see with muscles, and it shouldn't come to anybody's surprise that they're going to allow us to have movement, so things like locomotion and manipulation. They will also assist us with our posture and our body position, and it will also help stabilize the joints, and as the muscles contract, they also generate a lot of body heat, which can then be utilized for homeostasis and control of internal body temperature. All right, so let's go ahead and let's start taking a look at exactly what a muscle is made out of. And for this particular part of the chapter, we're going to concentrate on skeletal muscle. So a quick reminder on this slide is that when you look at muscles, you really are looking at an organ because it will have a mixture of things like the nerves, the blood supply, connective tissue sheets, and it will have different attachment points. In the skeletal muscle, please keep in mind that it is under conscious control. And also what we need to do is, as we are contracting the muscle, we're going to have to discuss the fact that it will rely on a lot of oxygen and nutrients to get this done. All right, now in a little bit, I'll show you an illustration of a skeletal muscle as a whole. And you're going to notice on that illustration that you have sheets, connective tissue sheets, layerings that are going to be labeled. You're going to see three of them, and they're going to be called the epimyosin, the perimyosin, and the endomyosin. And when you see these three terms being utilized, I want you to remember that they are a connective tissue sheet or a covering. And the way it's going to work is that the external one, the one that's going to be on top, is going to be called epimyosin. It is going to be composed of dense, irregular connective tissue. It's going to wrap itself around the entire muscle, so it's right on the surface, and it will blend in with the rest of the fascia or connective tissue within the organs as it's embedded within the body itself. Perimyosin is going to be your underneath level, and this is going to be lying underneath the epimyosin, and it's going to be surrounding what we call fascicles, which are going to be a group of muscle fibers. Now in a little bit this will all make sense when we take a look at the illustration, so hang in there with me. Your deepest connective tissue sheet will be called endomyosin. This one is going to be composed of fine areolar connective tissue, and this one will go ahead and encapsulate your muscle fibers. So from outer to inner, it's going to be epimyosin, perimyosin, and endomyosin. Let's take a look at the picture and make sure that we understand how all three of these will in essence lay with each other. Okay, so here you're looking at a skeletal muscle. So here's that bulky muscle that you see when you look at things like your biceps, your triceps, or like your calf muscle, your gastrocnemius muscle. And what this illustration is trying to show you is that the entire muscle organ is wrapped around by epimyosin. So as you can see, that is the external connective tissue coloring. And then it shows you that if we were to go ahead and zoom into the muscle, what we would see is that you have these little fascicles, these little smaller bundles, and each fascicle is wrapped around by perimyosin. Perimyosin, as it's wrapping around the fascicles, we see as we zoom in even further that the fascicle is composed of individual muscle fibers. And the individual muscle fibers will be wrapped around or protected by endomyosin. So the epimyosin is for the entire organ, the perimyosin is for the vesicle, and the endomyosin is going to be for the muscle fiber. Now, here is table 9.1, and what this one's going to do is it's going to show you the different structure and organizational levels of the skeletal muscle. And I want you to really take a good look at this table because it's going to explain to you the anatomy of the muscle as we start off from gross anatomy all the way down to microscopic anatomy. So the first one it highlights is muscle, which is what we're saying so easily. Muscle itself, remember, is the organ. So as you can see on the little picture, we have the epimyosin that's wrapping it around. And then what we see peeking out from the muscle organ is the little fascicle. And the fascicle is going to be our second row. And here we see that the fascicle is surrounded by the perimyosin, just as it was highlighted in the previous illustration. And it also says in the description that a fascicle is a discrete bundle of muscle cells. Now, those muscle cells, we're going to call them muscle fibers. So you can see it's sticking out. Through the vesicle, you see a little rod sticking out. It's labeled muscle fiber. And if you look down at the bottom of the table, that's going to be your third row. And you also notice that the muscle fiber, that's going to be the individual cell. It is an elongated, multinucleated cell. In this case, since we're looking at skeletal muscle, it will be striated. We're going to talk about that appearance in a little bit. And also notice how the muscle fiber is wrapped around by the endomyosin. So in fact, if you look all the way on the right-hand side of the table, it shows you how the connective tissue wrappings correspond to the muscle, the fascicle, and the muscle fiber. Now, when you hear me say muscle fiber, please keep in mind that's the individual muscle cell. And also what you're going to notice is that I'm going to say things like sarcolemma and sarcoplasm. The sarcolemma is basically the plasma membrane of the muscle cell. We don't call it plasma membrane as we do with all the other cells in the body. We call it a sarcolemma. So the sarcolemma is the plasma membrane of the muscle fiber. The sarcoplasma is the cytoplasm of the muscle fiber. And what we need to discuss is that within each muscle fibers, you're going to have different organelles that are going to be labeled, such as the sarcoplastic reticulum, the T-tubules, and the myofibrils. And our chores or our option for this is, as we're going through it, to definitely keep an eye on what does each of them contribute to the muscle fiber. All right, I really like this picture. This is figure 9.2, and it starts off by showing you a little histology picture on top, showing you your striations of your skeletal muscle, and it separates it to show you that those are what we like to call two different muscle fibers. So you can see the bracket on the side highlighting the fibers, and if you then look at picture B, you see that once again you have our muscle fiber drawn out, we have our sarcolemma, that has been labeled our plasma membrane, and we also see that there's another rod that sticks out of it, and those are going to be our myofibrils. Our myofibrils are where we're going to have our sarcomere, our myofilaments, and also where we're going to have our striations itself. So go ahead and take a look at C, and it basically says that if we take one of those myofibrils and we zoom in, what we see is we have this section called a sarcomere. The sarcomere is going to run from Z disc to Z discs. Now, the terminology that you see on your picture, Z disc, I band, A band, those are all different regions within the muscle fiber itself that will stain differently. And depending on the amount of stain and the shadow that it gives, it will be labeled by the individual letters. So we'll talk more about it in 1 second. But for now, go ahead and make sure that on the picture C, you can label the Z disc, and you can find the second Z disc. And if you look below, you're going to notice that it's been bracketed off, and they went ahead and they wrote down for you a sarcomere. The sarcomere will extend from one Z disc to the next, and it is the contractile unit of the muscle. It is the smallest part of the muscle that can contract. And the way it's going to contract is by getting your thin and your thick filaments to interact with each other. So in figure 9.2D, you see an enlargement of the sarcomere, and you see these little blue twisted rods labeled thin filaments, and you see these little larger red rods with little head parts sticking out. Those are going to be your thick filaments. And what we need to talk about is what are these filaments made out of, and also how do they interact with each other to get the muscle to actually contract and relax. All right, so let's take a look at these pictures just one more time, make sure that we understand everything. So here's just an enlargement of picture B, and it's a part of our muscle fiber. We see our little myofibrils sticking out, and I just kind of wrote down for you, it's a single muscle fiber, will contain thousands of these myofibrils. They're going to basically, it makes up a lot of the volume of the cell itself. And if we zoom in, we then start to see that we have our individual sarcomeres from one Z disc to the next. And we also start to see our striations. Our striations are going to be how we organize our thin and our thick filaments, thereby generating a dark and a light region. Hold on to that. I'm going to show it to you in 1 second. Dark and light region. All right, and as I mentioned to you before, a sarcomere is going to be the smallest contractile unit of the muscle. It's its functioning unit, and it will consist from one Z disc to the next. All right, so here it is zoomed in to the sarcomere, and what we need to do on this slide is we need to figure out what exactly are our myofilaments composed of. So what are the thin and the thick filaments made out of? So once again, if you start on top of the picture, you're going to notice the blue rods and you're going to notice the red rods. Notice how they're organized in a very organized pattern. So you have a blue, a red, a blue, a red, a blue, and a red. Because they are stacked up, so organized, that is then what creates the striations. So when you're looking at a muscle through the microscope and you're telling me, oh, this is a striated muscle, what you're really telling me is that the thick and the thin filaments are organized in a very dimensional order. So one after the other, a thick, a thin, a thick, a thin, very organized in its pathway. When you look at your thin filament, I want you to be able to tell me what it's actually made out of. So on your notes, if you look on the left-hand side, you notice that it's going to tell you that your thin filament is made out of actin. That's going to be the protein that it makes up. So all those little blue circles that you see intertwined with each other in the thin filaments, those are composed of a protein called actin. The thick filament, the red one, the larger one with the little head sticking out, that one is made out of myosin. And the myosin, if you look at the bottom of the thick filament illustration, it is basically two of them intertwined together, and it kind of looks like a golf club. And as you can see, as they're intertwined, they have a head region that sticks up. And the goal is to get the head region of the myosin to interact with the binding site or the active sites on the actin, on the thin filament. So we want to get these two to connect together, and that will be the starting point of our contraction. But we'll get to all that in just 1 second. For now, please know that the striations are due to the uniform pattern of the thin and the thick filaments. The sarcomere is the contractile unit and is composed of thick and thin filaments from one Z disc to the next. The thick filament is made out of myosin. The thin filament is made out of actin. And also what we're going to see is if we lie and we take a look at our sarcomere, what you're going to notice is that there are certain sections where there's a lot of overlap between the thick and the thin. And that will be made out of our dark region, our A region. And then we'll have a section where you have mostly thin by itself. That will be our light region. And it's really highlighted in this illustration right over here. So go ahead and take a look at your picture all the way on the top, and you can see the striations. You can see them lying on top of each other. I'm talking about the black and white picture. And then notice how you have the I band and you have the A band. The I band stands for light, and as you can see, it's where you have lots of thin filaments located. You can compare it to the picture at the bottom, the illustrated part, to see your thin filaments making up that particular section. The A band, which is your dark region, is mostly made out of thick filaments, and it will stain darker as in A band. A is for dark. It will stain darker because you not only have thick filaments there, you'll also have considerable amount of overlap between the thin filaments. Now, the dark black lines moving down the middle of the I bands, those are your Z discs. So once again, you notice all the way at the bottom of the illustration, from one Z disc to the next, they have labeled it as a sarcomere. And to keep the thin and the thick filaments in an organized pattern, we're going to have some regulatory proteins. So if you look over to the side, the left-hand side of the PowerPoint, you're going to notice some proteins with their function. The first one is going to be the CAP-Z. The CAP-Z will allow the thin filament to attach to the Z disc. Tropomodulin is a cap protein that you'll find at the end of the thin filament. Nebulum will go ahead and run parallel to the thin filament, allowing it to be stabilized. And titin will go ahead and hold the thick filament in place by allowing it to anchor onto the Z disc. Take a few seconds and take a look at the illustrated example that's right next to the text, and make sure that you can locate your CAP-Z. Remember, it's near the Z disc because it's holding on to the thin filament. The tropomodulin, that one's going to be at the end of your thin filament. Your nebulum, which is indicated by a green dotted line that runs the length of the thin filament. And then the titin is illustrated in blue. That one's going to be a thicker regulatory protein because it's going to allow to anchor the thick filament to the Z disc. Along the way, as we start diving in deeper and deeper into our skeletal muscle, you're also going to come across the term sarcoplastic reticulum. When you hear that, I want you to think about two things. I want you to think first about the fact that it's just basically smooth ER that you find in your myofibrils, so that you find in your muscle cells. And the second part is that sarcoplastic reticulum, or abbreviated as SR, will be a huge storage unit of intracellular calcium. So it will be able to hold on and store calcium inside the muscle cell, and it will release it when need be. So anytime you see sarcoplastic reticulum, SR, I want you to think smooth ER, and I also want you to think calcium storage and release inside the cell. You will also come across the term T-tubules. T-tubules are going to be little tubes that basically penetrate through the sarcolemma, the plasma membrane of the muscle, and they enter into the muscle fiber itself. Some books like to describe them as invaginations, infoldings, that basically create a pathway for the action potential that is found on the outside of the muscle cell to protrude to the inside of the muscle cell, allowing the individual myofilaments to get excitable. So as you can see on your PowerPoint, what I wrote for you is I wrote T-tubules are going to be voltage sensors because they're going to be able to detect the excitability that's happening on the outside, the sarcolemma, and they're able to then spread it to the inside of the cell because, like I said, they are infoldings or tubes that penetrate into the inside of the cell. Some pictures will go ahead and label a triad, and a triad will be an area on the myofibril where you will have your T-tubules and your sarcoplastic reticulum intertwined together. And usually it's called a triad because it will involve 2 SR regions and 1 T-tubule. Let me show you this on an illustration. All right, so here we see our little triad. So go ahead and look onto the right-hand side of your PowerPoint, and you're going to notice that you have your T-tubule drawn as a light blue line that basically runs the entire length of your muscle fiber. So that's going to be the infoldings. It's going to notice the excitability on the outside and allow it to penetrate to the inside of the cell. And then the darker colored blue that you can find on either side of the T-tubules, those have been labeled as the sarcoplastic reticulum. So those are your SR regions. And as you can see, you have 2 of those, and you have 1 T-tubule, making it a triad. And the SR region, that is where we're going to have lots and lots of intracellular calcium that's being stored. All right, so how exactly are we going to take this concept of thick and thin filament and sarcomere and sarcoplastic reticulum and T-tubules, and what exactly does that have to do with contracting a muscle? Well, for that, we're going to have to take a look at the sliding filament theory, or the sliding filament model of contraction. And it basically says that if you want to get your muscles to contract, you're going to have to create a cross bridge. And a cross bridge is going to be a physical attachment point between the head of the myosin and the binding site on the actin. So remind yourself that your thin filament is made out of actin and your thick filament is made out of myosin. And what you can do is you can go ahead and take a look at the illustration that's on the right hand side. And what you're going to see is that, remember how your thick filament has those little myosin heads that are sticking out? And they're coming in really close contact with that blue rod right above it. That's your thin filament made out of actin. And basically what the sliding filament of contraction says is that if you want your muscle to contract, the first thing we have to do is we have to make a cross bridge. We have to get the head of the myosin to physically connect with the actin binding site. And once they have that cross bridge, they're going to go ahead and do this thing called a power stroke that's going to allow them to basically glide over each other. And as they glide over each other, we're going to get the Z discs, the 2 black lines, from one Z disc to the next is a sarcomere, remember? So it's shown as those two black lines on our top pictures. We're going to get those Z discs to come closer and closer together. In fact, what you can do is you can compare the Z disc on the top picture to the Z disc on the bottom picture, and you're going to notice that they are closer together, and as they're coming closer together, you have more of an overlap between the thick and the thin filaments, and this is then our sliding filament theory. The muscle doesn't really shorten when it contracts, it simply slides its myofilaments over each other. And it will all start with the fact that we need to make a cross bridge, a physical connection between the myosin head and the actin binding site. So a physical connection between your thick and your thin filaments. All right, so let's take a look at this real quick, and then we're going to stop this part of the lecture and we're going to move on to the individual steps needed for the sliding filament. So let's make sure that so far we understand what's going on. So it says right here, it says in the sliding filament model, the thin filament needs to slide past the thick filament, causing the actin and the myosin to overlap. Remember that we're not changing the length, we're simply changing how much overlap they have together. And because this is a skeletal muscle system, we are going to have to rely on the nervous system to stimulate the muscle fibers. And as a response, we'll see that the myosin head will form the cross bridges, the physical connection that will start off the sliding process. And it will continue on, thereby shortening the Z disc for this area between one Z disc to another, allowing the sarcomere to, in essence, twitch or contract. All right, as we get to our last slide of this particular part of the presentation, I want to quickly show you an animation of a cross bridge and the sliding filament theory. So go ahead and take a look at the left-hand side of your screen, and you're going to see a very simple animation of a thick and a thin filament interacting with each other. The thick filament is indicated by the orange section, and you can see the little heads sticking out at the top and at the bottom. And the thin filament is shown by the little black rods. And what it's showing you is how the head of the myosin of the thick filament is making physical contact with the thin filament, and that, of course, is the binding sites of the actin. And as they make that physical contact, that is going to be the formation of their cross bridges, and as they start sliding, we then see that as the muscle contracting and relaxing. You can also see that happening on the right-hand side of the screen if you take a look at the illustrated example where you have the Z discs labeled, and notice how in picture 1 compared to picture 2, the distance between the Z disc is different. If it's a relaxed muscle, the Z discs are further apart from each other. If it's a contracted muscle or a contracted sarcomere, the Z discs are closer to each other. And this all has to do with the fact that if you want to contract the muscle, you're going to have to form that physical connection, that cross bridge, and you're going to have to increase the amount that the thick and the thin filaments are sliding or overlapping with each other. Now, as you're looking at these illustrations, hopefully a question that comes to mind is how do we prevent cross bridges from forming if we want the muscle to be relaxed? How do we prevent the myosin head from interacting with the actin binding site if we don't want the muscle to form a cross bridge, if we don't want the muscle to go ahead and start contracting? Well, for that, we're going to have to take a look at what troponin and tropomyosin do for the whole process. So let's take a look at this little illustration that's going to pop up. And what we see here is we see our little blue beads. Those are our actin binding sites. That is what makes up our thin filament. And what we see happening is that on the illustration, they've also labeled tropomyosin and troponin. And the tropomyosin and troponin complex will work together, and what they'll do is they will go ahead and shield or cover the binding sites of the actin when you don't want your muscle to contract. So when the muscle is relaxed, the tropomyosin hides the binding sites. You can see it being illustrated as like a string-like protein, and it's covering the binding sites. And if you want your muscles to contract, then we need to slide the tropomyosin out of the way so that the head of the myosin can get in direct contact with the actin binding site. And the protein that's going to be able to slide the tropomyosin out of the way is going to be troponin. So they have to work together, and we'll talk about in our next presentation what exactly it takes to get the troponin to move the tropomyosin so that the head of the myosin can bind with the active site on your actin or your thin filament.